Hello everyone, in this video we're going to talk about our guy Mendel, very exciting, and then we're going to talk about some of the background knowledge for Punnett square, some of the vocabulary that you need to know very clearly because we're going to keep on using these vocabulary and they're going to show up on the test because they're really important. Okay, so starting with something called the blending hypothesis, it kind of looks like this. You're blending two paints, but really in the past people thought that genetics works like the blending process where you just take some genes from mom, take some genes from dad, and you blend it together, then you get a child. Well, it sort of works that way, but it really doesn't. You don't get a mixture of the two, right? Okay. I mean, you do scramble the genes together, but you don't really blend the two together. It's not as simple as that. So Gregor Mendel was the person who figured out that the blending hypothesis is untrue. You don't get a yellow-green pea from yellow and green pea. You would get either yellow or green usually, and not a mixture of the two colors. Simplest way to explain this. So Mendel, here's just some background knowledge, but basically you should know that he is kind of the godfather for genetics. He kind of made some groundbreaking discoveries on how genetics works. So let's start with Mendel's piece. Mendel looked at 7 traits or characteristics of pea plants. So you should know that pea plants are the ones that Mendel worked with, and there are some specific characters that he looked at. But he focused on one trait at a time. So if you were to look at a human, right, you can just look at me. There are so many things that are going on. It would be very complicated for you to figure out anything by doing experiments on the spy. But if you were to look at pea plants, which don't have as many traits, you can focus on one trait at a time and then kind of see what's happening. So for example, you can see round peas versus wrinkled pea. That's one trait that we can focus on. Remember, one gene, one trait. So there is a pea-shaped coding gene that codes for the shape of the pea, right? And they are at the same locus, and we'll talk about what locus are. And then there's the pea color, yellow versus green, that's coded by one gene. Just turned out this way that there are some traits that are coded by multiple genes, but in this case, all of the traits that we happen to look at are one gene, one trait. We can have purple versus white flowers, one gene, one trait. All right? So here's blending hypothesis again. Here's Mendel with, have no idea what he's holding. Anyway, pea plants. Mendel also used mathematics to explain some of his results. So as you know, when we do Punnett squares, we talk about ratios, we talk about percentages, we talk about the predictions of what the offspring is going to look like. So Mendel did all of that. That's how we know how to do it. He predicted the concept of genes. Genes occur in pairs. So if you remember homologous chromosomes, right? Homologous chromosomes, so this is the important one. Let's write it down. So we did this in class as well, but a refresher. Whenever you have a cell that's diploid, yeah, that works. Whenever you have a cell that's diploid, that means it's 2N. That means there are homologous chromosomes. That means there are also, well, let's start with somatic cells. So somatic cells are any of your body cells that's not sperms or eggs. So that's most of your cells, right? So for pea plants, anything that's not the pollen or the egg in the flowers are somatic cells. So even pea plants have somatic cells. Whoa, mind-blowing. And then homologous chromosomes, that also means you have 2 alleles of the same gene. So remember, one gene codes for one trait, and 2 alleles of the same gene means 2 versions or 2 types of the same gene. So what does that mean? Remember the marker example, right? So let's say we have 2 homologous chromosomes there. It says most of the time the genes occur in pairs. So here we go. On these 2 homologous chromosomes, we could have eye color black, eye color red. They occur in pairs, but because they're both coding for eye color, that means they're the same gene. But because one is coded for black, one is coded for red, they're 2 versions of the same gene. So we call this part one allele, the black allele for the eye color gene, right here, the red allele for the eye color gene. So you really wanted to get the wordings of these descriptions right. Okay. So here's all the vocabularies that we're going to be looking at today. So first off, a gene is a sequence of DNA coding for a protein or a trait. Okay. A genome is the entire set of genes in an organism. So for a human, we would have 23 pairs of genes. Well, that was so wrong. Redo. 23 pairs of chromosomes as our genome. Okay. Well, that's for a person without a chromosomal disease. You would have 23 pairs of chromosomes. So this is a dog's genome, which is 39 pairs of chromosomes. This makes up a dog's genome. So all of the genes, every single gene on every single chromosome, if you're considering all of them, that's your genome. Now, alleles and locus. So let's first focus on locus. Locus is very similar to the word location, so that makes sense. A locus is where a gene is located on a chromosome. So the locus of that eye color gene right there, or earlier, is going to be right here. So that would be the locus of the eye color gene. That's where the eye color gene is located on this chromosome. So another gene, let's say hair color, would have definitely a different locus compared to the eye color gene. So the hair color gene might be right here. Okay. So the important thing is you need to know what a locus is where a gene is located. So for the same gene, they should be located at the same locus on a pair of homologous chromosomes. So the locus for the black eye color gene should be the same as the locus for the red eye color gene, as you can see. Now, the second one, allele. Allele, this definition right here is a little complicated. You can read it, but it tells you something, right? So alleles are at the same locus. Again, you know that's true. We have our alleles, 2 alleles of the eye color gene. They are located at the same position on the homologous chromosomes. That describes where the allele is. And you should know that they cover the same trait, right? Eye color, that's right. But because alleles could be the same version or different versions of the gene, you will say alleles are 2 versions or versions of the gene, right? So you could have for every allele, right here. For every gene, so let's say gene, eye color gene, right? So that's one gene. We can use any color as we want, but let's just say we use A. We could have the dominant allele, and we could also have the recessive allele. And we express it by using either a capitalized letter or a lowercase letter. Dominant allele, recessive allele. There's only one letter because one allele is one version of the gene located on one chromosome. And when you have 2 letters together, let's say you have 2 letters together, right? These are the only 3 combinations that you can come up with. These are for somatic cells. These are the possible gametes that can be made from each one of those parents. Okay? So moving on. We'll talk more about that when we talk about Punnett square. But here we can have, for eye color, we have 2 different alleles, 2 alleles, 2 alleles, 2 alleles. All right, so if you think back to meiosis, this is why meiosis is so important, because we can't talk about Punnett square or anything without knowing how meiosis works. So for meiosis, we start with a parent cell, right? A parent cell, because they're homologous chromosomes, you would have 2 alleles of the same genes, as is written right here. So you should be able to associate each one of these words with, each one of these other words. And then the next one is gametes. Gametes would have 1 allele of the gene because gametes are all haploid. So as you can see, these 2, each one of them could be a gamete because each gamete carries 1 allele of the gene. And then offspring, because offspring puts 2 gametes together, we would have 2 alleles of the same gene as well. Okay, but this is right here, we're only looking at 1 trait, but you can also look at multiple traits. Let's say we look at A, B, C, and D together, right? The parent genotype could be big A, little a, big B, C, C, and round space. Let's say this is the parent, right? But then the gametes that can be made could be big A, big B, big C, or it could be little a, little b, little c, or it could be little a, and then it could also be this. But either way, within each one of these gametes, you don't have to know how to do this, but what I'm showing you here is that within a parent cell, or within a parent body, or the offspring body, you would have two allele of the same gene. So for each letter, you have two of them, right? Whether it's big or small. But within a gamete, right here, you would only have one allele of each gene, right? See? Now you only have one copy of each letter, meaning you now only have one version of the gene, not two of them acting together, like the parent or the offspring, okay? So dominant versus recessive. Dominant versus recessive, we're always talking about, well, we could talk about it in terms of genotype, and we can talk about them as phenotype. So we can have dominant alleles and dominant alleles coming together. In this case, it will be big A, which means it's going to show the dominant trait. Okay? So there's a difference between genotype and phenotype. Genotypes are the letters. Phenotypes are the trait that you can see. So if you were to have one dominant, one recessive allele, as this case, then you will still see the dominant trait. So in this case, you'll see the purple color because that's what makes something dominant. It covers up the recessive trait. The only time that you can see the recessive trait at all is when you have two versions or two alleles that are both recessive. Two recessive alleles will give you the recessive trait. That's the only time that this will work, okay? So dominant, you can associate it as a capitalized letter, such as big A or big B, but when you say dominant allele, it has to be one letter. One allele, one letter, okay? Recessive allele will be the lowercase letter, but again, that's one letter. But when you look at a parent, remember we said parent has to have two alleles, so the parent could be this, or this, or this, but we give it different names. Oh, wait. So this right here shows you, this is a dominant allele, dominant recessive allele, recessive trait, dominant trait. You wouldn't know which one is which unless I tell you, but brown is dominant over blue. Okay, homozygous, heterozygous. Homozygous means you have two identical alleles, one from each parent because everybody has a parent at some point. So right here, big A, or on the parapoint, big B, that will be homozygous, but little b also is called homozygous because they have identical alleles, one from each parent. The two alleles are always one from each parent. Okay, that's how this works. Heterozygous have two different alleles, so the one in the middle is called heterozygous. So the first one, big A, is going to be called homozygous dominant because it has two dominant alleles, and it's going to show the dominant trait. So big A will be the genotype, the dominant trait, in this case purple flower, will be the phenotype. All right, big A, little a will be heterozygous. It's still going to show the dominant, it has one dominant allele, one recessive allele, but it's going to show the dominant trait. And then we have homozygous recessive, little a, which shows us the recessive trait, and it has two recessive alleles. So little a will be the genotype, white flower will be the phenotype. This is just the same idea. You can take a look and see if you understand that. Again, genotype versus phenotype. Genotypes are the letters. Phenotypes are the trait that you can see, the actual color, the actual height of a person. All of that, okay? So take a look at this. A few more things. Monohybrid cross, meaning you're only looking at one trait. So if we're crossing a parent with big A, genotype, and big A, little a, genotype, that will be a monohybrid cross. If we're crossing two parents, you are looking at three traits at the same time, then that will not be a monohybrid cross. The next one that we can talk about is the parent generation, first generation, second generation. So let's say your grandparents are the parental generation. Your parents' generation will be F1, and then your generation will be F2. So it goes like P, F1, F2, and you can look at how each generation crosses with each other and what is the result. Okay, I hope. Okay, I hope this makes a lot of sense. And if you don't, I'm at the library in the morning, and please come and ask questions. Have a good one.